little bit of parenchyma to the inside. And note that this cork cambium here is at a more interior position at that section than it is above. Above there, the cork cambium is in the cortex, but here we can see the cork cambium is formed back into the secondary phloem, or into the primary phloem. So what happens is that the cork cambium is eventually destroyed as the stem expands. Everything to the outside of the vascular cambium is under this expansion stress that eventually destroys that tissue. And so we eventually have the need for a replacement cork cambium. So cork cambia are always forming further back into the stem as the stem expands in girth. And eventually the cork cambium is going to be forming back into the secondary phloem, so that everything to the outside of the cork cambium is going to be either cork cells, parenchyma, or secondary phloem. And everything else is eventually sloughed off. So there's constant sloughing off of tissue. And if you look in this longitudinal section, you can see the primary xylem here is in the same position all the way up in terms of basically this line is vertical. Whereas you can see that the secondary xylem, this lighter blue, gets narrower and narrower as we go upward. So as we come down from the top, this is a longer and longer period of secondary growth that initiated at a higher point, and we have more wood lower on the stem. And you can see the vascular cambium is also moving out this red line, and out to the outside here then we have phloem and periderm. Periderm is the term used for the tissue produced from the cork cambium. So that's basically how a stem gets larger, and the different this is just showing you the different layers that I just discussed, and this is a good slide for studying this particular diagram. All right, so you can imagine if you're replacing the epidermis with all this suberized cork tissue on an old stem, it's difficult for any gas exchange to be able to occur in the stem. And there is the need for gas exchange in stems, and the respiration has to occur in these cells, requires oxygen. And the way that woody plants get around this is that they have these regions, these light-colored regions called lenticels, and you've no doubt noticed these on branches of plants. These are basically loose arrays of cells that are very loosely packed that allow for gas exchange, similar to a stomata, to the stomata of the epidermis, but this is instead periderm. And the shape and size, color of these lenticels varies a lot between different species of woody plants and can be really taxonomically useful for identifying woody plants at times of year when they've lost their leaves and a lot of their other characteristics. So lenticels are often pretty diagnostic for identification of different taxa. All right, so in later secondary growth, as in this cross section you can see here, you can see that the wood is much more extensive than the bark. And there are a couple of reasons for that. First off, that little cartoon I showed that showed this alternating production of one xylem cell to the inside, alternating with a phloem cell to the outside, that's not completely accurate. There are more divisions to the interior than there are to the exterior. But also important is the fact that everything to the outside of the vascular cambium is eventually sloughed off. We have that expansion of the tissues there, that shearing. And so phloem and as well as periderm is constantly getting sloughed off as the stem expands. It's being replaced by more secondary phloem and more periderm from the cork cambium. So those two factors together result in the bark being relatively thin compared to the wood. The wood's not under any pressure. It's being produced and left behind. It's not under any kind of compression force. All right, so there are a couple of different kinds of wood that are important to understand, especially in trees that get pretty sizable. You've probably heard of heartwood and sapwood, and you might even realize that heartwood is darker in color than sapwood typically. You can see the large amount of heartwood in this cross section compared to the light-colored sapwood. The sapwood, really secondary xylem only functions in water conduction for a relatively short number of years compared to the life of the plant overall. And after a certain point, the center wood, of course, is the oldest, and after a number of years, the tracheary elements, the tracheids and vessels, will no longer function as conduits for water transport, whereas they do in the sapwood. It's called sapwood in reference to the xylem sap, which is just the water and dissolved minerals that are moved through the xylem. The heartwood is a place where the opening, the lumen of those tracheary elements gets filled with secondary compounds, resins and such, and it becomes important for structural support of the tree. It's very tough wood because of the being a depository for these secondary compounds. It also tends to be pretty impervious, or relatively impervious to insect damage because of that, and it's the most valuable lumber in timber trees. The heartwood is the prized lumber typically. I mean, it's the bulk of the wood in any case. And in temperate trees, that is trees outside the tropics and into the boreal regions where you have seasonal growth, typically the wood will have this ring sort of pattern. You've no doubt heard of tree rings. This isn't something we see in tropical trees, and the reason is because growth is possible year-round, at least in tropical environments where there isn't any seasonality. In areas with seasonal growth, we typically have early wood looking relatively light in coloration because the tracheary elements are relatively broad in diameter. Early in the season, resources are more abundant, and the tracheary elements are typically larger. And late in the season, the late wood is denser, and the tracheary elements, the xylem and vessel, or the vessels and tracheids are narrower in diameter, and the wood is denser overall. And taken together, this early and late wood from a given year then forms an annual increment tree ring that allows dendrochronologists, people that study climate change and such, to reconstruct past climates, because depending on how dry or cold the conditions were, the characteristics of these tree rings will differ quite a lot in terms of their width and composition. So there's a lot that's been learned about climate since the Pleistocene by looking at the quaternary record, by looking at tree rings, not just living trees, but also dead trees that have where the wood is well preserved. So these tree ring records have been really important. All right, so monocots sometimes can achieve the dimensions of trees as well, even though they can't produce wood. And there are multiple ways that they can do this, but we're only going to talk about one. And this involves some of the most conspicuous monocots, the palms. And some palms can have fairly large diameter trunks. They're definitely trees, but they don't have any wood. And if you look at a cross section of a palm stem, you can see up there those little dark circular regions. Those are vascular bundles scattered throughout that stem. Most of that's ground tissue, that stem cross section. And even though there isn't wood there, the xylary elements, the tracheids and the vessel elements, have strongly lignified secondary walls. So there is some strength that the xylem imparts to that stem. But it gets to be so broad because the shoot apical meristem is incredibly broad in a palm. So from the time that the stem tissue is first formed, it's the same diameter as it is later. So they have a very straight trunk. And Same goes with bamboo, which is a type of grass. And if you look at a bamboo, what's so-called bamboo wood, under magnification, you can see the vascular bundles scattered in the ground tissue. It's definitely not wood. And so here you can see some bamboo on the left, palm in the middle, and a hardwood eudicot on the right. And the stem width is, as you can see in the bamboo especially, more or less uniform from base to summit. In the palm, that's true. There's a minor bit of flare there at the very base, but that's also due to primary growth. The hardwood has a very, very narrowly triangular type of stem from the tip downward, which is due to the region at the base of the trunk has had a longer period of time for secondary growth than regions above it. So it's just the time duration for secondary growth. All right, so here's a few questions just to kind of test your understanding of secondary growth. So we hammer a sign into a tree a couple meters from its base, and the tree elongates a meter each year, starting out at 10 meters tall. How far up and how high will the sign be after 10 years? The same, right? It's just going to be 2 meters tall, so it's a trick question. I don't ask questions like this on exams, but that's a trick question. So if the nail holding the sign is nailed in as far as the secondary xylem, all the way into the secondary xylem, how high will the sign be? It's the same again, right? It's in the secondary xylem. In fact, that's what happened here in this case. It was nailed into the secondary xylem, and we've had this expansion of the tissue to the outside. The sign's kind of held in place in terms of its position, and the tree's growing around it. But if the nails holding the sign are nailed in only as far as the secondary phloem, how high will the sign be after 10 years? Right. It could be sloughed off because the bark is constantly being sloughed off. And remember, the bark is everything to the outside of the vascular cambium. All right, so there's another set consequences of secondary growth that are important for understanding woody plants. So if you were to basically remove a continuous strip of bark all the way around the tree, and it can only be just an inch of bark all the way around. Remember, you're removing everything down to the vascular cambium. Imagine what the consequences of that are going to be for the tree. It can't produce any more bark. Its vascular cambium's been killed by doing this. So it has wood, all the wood's still in place. And remember that the conduction of water involves these non-living tracheids and vessels mostly. I mean, that's those are the cells involved in the transport of water. It's not an energy-requiring type of transport. So water can be free to move up through the roots to the upper shoot above that point of what we call girdling the tree, removing a continuous strip all the way around the vascular cambium of the bark. But the photosynthate, moving down from the leaves, reaches the girdle, and then it's just going to accumulate there at the top, and it can't reach the roots. So the roots are dependent on the photosynthate from the leaves, and the roots are going to starve to death. So that's what kills plants when they lose a continuous strip of bark all the way around them due to accident or somebody intentionally girdling the tree. Okay, so, all right, so in the last few minutes here, let's talk a bit about the root anatomy and morphology. In a eudicot, the radical persists, becomes the taproot. We have branching from that to produce lateral roots that are identical to the main root in their anatomy and morphology, just as branches or axillary shoots are identical to the main shoot that produces them. In monocots, though, we have a fibrous root system. I mentioned earlier, these are adventitious roots. Adventitious just meaning any structure that's formed in a position other than where it's normally formed. And these are formed from the base of the stem, so the taproot dies away quickly, replaced by fibrous roots, which are often really shallow, in contrast to the taproot system of eudicots and other dicots. So primary elongating growth is all we're going to talk about in roots, secondary growth similar to the shoot system. And note that the apical meristem is a bit back from the tip of the root itself, that little zone that's delimited by dotted lines there, the embryonic region of the root tip. And you can see the zone of cell division runs from about here up to it overlaps with the zone of elongation a bit. And back from that point, no more cell division, but there's more elongation. And then the zone of elongation extends a bit into the zone of differentiation, where we have different cell types becoming differentiated from one another. And you can see root hairs forming in that zone of differentiation, where we have, those are basically extensions of epidermal cells that increase the surface area of those cells and enhance water uptake. Okay, but also notice that there's this thing called a root cap here that's more apical than the apical meristem. So this apical meristem is not only mostly undergoing parallel divisions that are parallel to the summit of that apical meristem back from the tip, they're also undergoing cell divisions toward the tip that produce this protective root cap that shields the apical meristem from abrasion as the root is pushed through the soil by the growth of the root. And the root cap also produces slime, this mucilage that lubricates the root tip and allows it to push through the soil without so much damage. And the root cap's constantly being replaced, the cells there as they're being sloughed off. The root tip also serves a function in positive gravitropism of the roots. So roots grow downward in response to settling or tension inside the cell. The current thinking is that the plastids of the roots, remember the plastids in the leaves are differentiated for photosynthesis, the chloroplasts. Plastids in roots that are differentiated for storage of starch are called statoliths, and they will settle on the lower side of the leaf in response to, on the lower side of the root cell in response to gravity. And that then results in an interplay with the growth hormone auxin, which results in more growth on the upper side of the root cell than on the side that's lower. And that then results in growth of the root downward, as in this little video here. So the growth gradually becomes vertically downward. All right, so let's look now at the anatomy of a eudicot root in comparison with a stem. So the epidermis is the same, although it has root hairs. So the hairs on the stem and leaves have various functions. The stems, the extensions of the epidermis and the roots serve the function of absorption of water and inorganic nutrients. They increase the surface area. It's passive absorption of water in the case of water. Cortex is again present between the epidermis and the vascular cylinder. There's a vascular cylinder, but the innermost layer of the cortex is called the endodermis, and this is involved in limiting prevents the soil solution from diffusing into the vascular cylinder and contaminating it. We'll talk about that shortly. That's the innermost layer of the cortex, the endodermis. The outermost layer of the vascular cylinder is called the pericycle, and it's where lateral roots are formed, the branching roots. The xylem is at the core of the root, and it's a star-shaped arrangement of the xylem with the phloem in between the arms of the star in case of a eudicot. All right. And here you can see it in a real cross-section. Most of this is cortex, but here we can see the vascular cylinder with the vessels there with the red staining, secondary walls, the large lumens openings, and the phloem in between the arms of that star. We can also see the endodermis here, those cells that are red staining, that's from the suberin, a waxy substance in the cell wall that prevents water from diffusing through the cell walls into the vascular cylinder and contaminating it. We'll get to that function shortly. The formation of branches of the roots, root branches or lateral roots, is not like in stems where we have an axillary bud that can be activated and already has a vascular connection to the main vasculature of the stem. There are no buds in roots. And so we actually have the lateral roots forming from the vascular cylinder itself and growing out, basically rupturing the tissue of the cortex and epidermis as it emerges. But the endodermis, that innermost layer of the cortex, is continuous between the vascular cylinder of the main axis of the root and that branch. So the vascular tissue is protected from getting contaminated by soil solution. There are various modifications, and we'll talk about that in a moment, or actually next time. There are various modifications of roots for different purposes other than absorption or anchorage. There are prop roots that can support top-heavy plants like monocots that don't have secondary growth. These can form from nodes, as you can see here in the case of this corn plant. There are storage roots like a carrot or a beet or a turnip that store carbohydrates in biennials that flower in the second year, and those resources, the water and nutrients, will be used for growth in the next year. And there are buttress roots, like in the case of a fig or ceiba, that are basically just a type of prop root in tropical trees that help to support these large trunks. All right, and we'll finish up with roots next time, and that'll be the end of the material near the beginning of the lecture for the midterm. So I hope you can all enjoy the holiday weekend and get some rest, and see you back here Monday.